So we are talking about the third step of glucose oxidation. So remember, everything that's happening in cell respiration is to oxidize glucose, to take it back to carbon dioxide, whereas in photosynthesis we had carbon dioxide and we reduced it to make glucose. Now I'm abbreviating this step ETC. ETC stands for Electron Transport Chain, and I mentioned that earlier. Now, unless I'm specifically asking you what is the name of the third step, you are free to use the abbreviation ETC if you're talking about it or mentioning it as part of an answer that you're giving to a question. Now, what is the electron transport chain? Well, it is a series of oxidation reduction reactions. That means that something will get oxidized, and because of that, some other molecule will be reduced. Now I'm going to remind you that in the context of our course, you should look for hydrogen. Something that has hydrogen could be oxidized. Something that doesn't have hydrogen could be reduced. Now, where is this happening? It's happening on the cristae. So if I look at a mitochondria, it's got the wavy line on the inside. That is the cristae. Now, basically what is happening here is that molecules that have picked up hydrogen are going to bring it here. So we saw in our previous lessons that there are molecules that pick up hydrogen. NAD picks up hydrogen to become NADH. FAD picks up hydrogen to become FADH2. So the hydrogens that are being transferred are coming from our transport molecules. That's where I'm getting them from. So these molecules that I've made throughout the other steps of cellular respiration are all coming to the electron transport chain to transfer their hydrogens. Now we're transferring them to things called cytochromes or electron acceptors. They have actual names, which I would not expect for you to know. So we're just going to talk about acceptors. These acceptors are really just proteins that are incorporated into the membrane. So that's how you can refer to them if you'd like to say what they are. Now, what do? As they're transferred from one protein or one acceptor to the next, energy is being released. And what does that really mean? As hydrogen are moved, ATP is produced. We already learned about the process of chemiosmosis and about the structure ATP synthase. That's how I'm getting ATP. Hydrogen ions are being pumped to one side of the membrane, and as they come back through, their movement activates ATP synthase and helps us produce molecules of ATP. So hydrogen are bouncing along from one protein to the next. The last acceptor, and this is a really key point because it's the last one. Another way that I could say last would be terminal. So the terminal or last electron acceptor is the strongest one. It's oxygen. And this is where I get water. So if you might remember in photosynthesis, when I broke apart a molecule of water, I got oxygen. In cell respiration, when I add some hydrogen to oxygen, I get water. So I'm putting things back the way they were in photosynthesis. Now the next point explains why oxygen is necessary. If the cell lacks oxygen, the ETC doesn't work. It can't work if the last acceptor is not there. It makes the whole rest of the system back up. If that doesn't work, neither does the Krebs cycle. So that is why, even though oxygen is not officially part of the Krebs cycle won't work if oxygen is not present. And that's because the products of the Krebs cycle have to go to the electron transport chain. If it's not working, the Krebs cycle gets backed up as well. Now these are our transport molecules. NADH is the first one. It drops its hydrogen off at the first cytochrome. FADH drops its hydrogen off at the second cytochrome. Now these cytochromes, remember, are just proteins in the membrane. And there are 3 total. So the one that drops its hydrogen off at the first cytochrome has 3 spots to move. It makes 3 ATPs. The one that drops its hydrogen at the second cytochrome only has 2 spots to move, so it only makes 2 ATPs. Once these 2 molecules have dropped their hydrogen off, this is what's left. NAD positive and FAD positive. They don't have their hydrogen anymore. And they can go back to the Krebs cycle to pick more up. So the Krebs cycle and the electron transport chain go hand in hand. One really does not function without the other. So I'm just going to pause here for a second. So on page 188 of your textbook, the way that the electron transport chain is represented is by a set of stairs. So at the bottom of that page, you've got a set of stairs, and it's showing some things coming in and some things going out. So the key idea is that you understand that each of these steps represents a protein, some protein in the membrane, in the cristae. So when I said cytochromes were proteins, that's what I'm referring to. These are the acceptors. Now it doesn't actually look like a staircase. This is not its actual appearance, but this is a way that you can think about it. NADH, like we said, comes to the first cytochrome and drops off its hydrogen. What's left? NAD positive. So what happened right here? NADH got oxidized. It lost its hydrogen. Now, since this hydrogen has to move 1, 2, 3 times to get all the way down to oxygen, which is the last acceptor after it's gone through the proteins, each time it moves, eventually an ATP is going to get made. It's not a direct process. So each of these little dotted arrows represents chemiosmosis making 1 ATP. So we've simplified it. We're just trying to give a visual representation of what are the steps that are happening. Now, chemiosmosis happens through ATP synthase, which is another structure, but just so that you can see where I'm getting all of the ATP from. Now, my other molecule, FADH2, like it shows in your textbook, comes in a little later. Now, I like to look at this and say, oh, look, FADH2 has a 2 on it. That reminds me that it comes to the second cytochrome and that I only get 2 ATP from it. So it's a good little trick to remember. It drops off hydrogen here and becomes FADH positive. Since this hydrogen only has 2 steps to move, I'm only going to get 2 ATP from it. So this is the idea that's represented in your textbook in a very simplified format so that you can understand that there are 3 acceptors that are proteins, and then there's oxygen. NADH comes to the first one. That's why it makes more ATP than FADH2, because it's only coming in at the second one. The other thing that you still have to know is what chemiosmosis is. So I'm going to show you a more detailed picture now. If you think that you'd like this picture, you have some choices. Google it, and you will find a million pictures that look just like it. Go on my website, print it out from the package of notes that's there. But let's not spend 4 hours with everyone trying to copy this incredibly detailed picture down right now. So I'd just like to show you what this looks like if I were to put it in the actual mitochondria. Is that a question that you have? It's dropping 2 hydrogens, but the fact that it's dropping 2 isn't something that we're going to focus on. It's not making a difference in the ATP production. So you're right, it does have to drop 2. I'm illustrating just one there so that it's comparable to what's happening with the other one. So this is what it actually resembles if I was to look at a mitochondria. So I'm going to point out some things that are happening. This is ATP synthase. It's a structure that is found in membranes. It's found in the mitochondria. It's also found in the chloroplast. So let's not forget, this is not a structure that is unique to the mitochondria. Its job is to produce ATP. As we already know, hydrogen has to move through it. ATP will get made. So that's how I'm getting ATP. Now what's happening? On the top, we're illustrating what NADH does. Notice that there is 1, 2, 3 protein acceptors that hydrogen and that movement is going to, or that energy is going to travel through. Every time it moves, the result is that hydrogen is pumped out so that it can go over here and move through ATP synthase. So on the little staircase, it kind of looks like everything is all together, but so that we could get a realistic representation, the hydrogens have to go off somewhere else to make ATP. Since this is happening 3 times, well, I get 3 ATPs. And then waiting at the very end is oxygen. And that's how I end up making water. So this is all happening on a membrane. Remember that a membrane looks like that in a picture. The little round parts are phosphates, the inside parts are lipids. The bottom illustrates FADH2, and it looks very similar, except that I don't have as many places that I'm going to move. So if I look at FADH2, I only have these 2 big guys. Yes, there are other little molecules along the way, but I just have those 2 big ones there. It's like I skipped over the first part. And so I'm only going to get hydrogen 1, 2 times. And that's how I'm going to produce ATP. Just like with NADH, oxygen is waiting around at the end. And this is how we are going to make ATP, and this is how we're going to make water. The Krebs cycle is happening inside the matrix. So inside this membrane, that's where the Krebs cycle is. So that is where FADH came from. That's where FAD goes back to. That's where NADH came from. That's where NAD positive goes back to. So that transport molecule is constantly moving between the electron transport chain and the Krebs cycle. Would anyone like to ask any questions about the more realistic picture of how the electron transport chain works? So then I'm going to direct you to take out your practice quest and flip it to the last page, because the question that it asks you there, we're going to answer right now. Now, the question that I'm asking you at the end of this practice quest is to illustrate how the first steps of cell respiration are used in the electron transport chain to produce ATP and water, which are kind of the end results of that particular cycle. It says be specific, use numbers, say where things are happening. So basically, I'm asking for a summary of the molecules that give me energy so that I could figure out, hey, how do I get 36 ATPs, which are part of the cell respiration reaction? So I'm going to focus on one glucose molecule. That means that some of these things have to happen twice. So even though technically there are 4 steps, you might remember that I said the other day the link kind of goes with the Krebs cycle. So technically there are 4 things that are happening. What I'm illustrating to you are all of the molecules that either are already ATP or that will be used to produce ATP in the long run. So when I talk about glycolysis, notice I'm not focusing on the carbon compounds. We already know that CO2 comes out in the Krebs cycle. I'm focusing on the energy compounds. Glycolysis produces 2 NADH and 2 net ATP. You might recall yesterday, you actually get 4, but you had to use 2. Now, if I take this idea a little bit further, those 2 NADHs are going to eventually make ATP for me. So at the bottom, I'm reminding you what the electron transport chain does. For every 1 NADH, I get 3 ATPs. So that means that those 2 NADHs will have made, in the long run, 6 ATPs. Now these 2 ATPs are already ATP, so I don't need to do anything to convert them to it. So the next step is the link. Just like with glycolysis, I'm going to get, in the long run, 6 ATP. And that's because of this point once again. Every NADH will help make 3 ATPs in the electron transport chain. Now the Krebs cycle, I'm illustrating as if it has happened twice. And that's because it has to happen twice for one glucose molecule. So in reality, one cycle of the Krebs cycle produces half of this. I'm showing what happens when it happens twice, so that we always have the overall picture. I've already got 2 ATPs, so I don't need to convert those. Then I have 6 NADHs. Each NADH is going to give me 3 ATPs. 6 times 3 is 18. I also have 2 FADH2s. And as we illustrate down here, the electron transport chain converts FADH2 to ATP 2 to 1. So for every 1 FADH2, I get 2 ATPs. So if we add this up, what do we have? 6 plus 2 is 8. Plus another 6 is 14. Plus 2 is 16. plus 18 is 34, plus 4 is 38. And then you'll say, didn't you say that there were 36? That adds up wrong. And I will say, of course. I said there were 36, so there must be something else that is happening. So here's the kicker. If you are a multicellular organism, 2 additional ATP are used up. The reason they're used up is because there are membranes that things have to cross. For example, NADH that's made in glycolysis has to get across the membrane of the mitochondria. This means that a unicellular organism could produce more ATP than a multicellular organism. But for most organisms, and in particular for humans, which is what we are, 36 ATP is the total. Now the last thing I want to just mention, and then I'll see if there are questions, because the question on your quest asked you where these things happen. I'm going to repeat that. Glycolysis happens in the cytoplasm. The link in the Krebs cycle happen in the matrix, and the electron transport chain happens on the cristae.